Hello friends, today we are going to study about composition, structure and function of proteins. In that we are starting with amino acids. What are amino acids? Amino acids are the building block for proteins, or in other words we can say proteins are the polymers of amino acid. Here all the amino acids are joined by a covalent bond, and after joining these amino acids are known as amino acid residues. Why? Because during the formation of covalent bond, H2O is released. Therefore, the amino acids after joining are known as amino acids residues. The first amino acid discovered is asparagine, and the last amino acid discovered was threonine. All proteins are composed of 20 standard amino acids. Most of the protein contains most of these 20 amino acids, if not all 20. So, amino acids are the building blocks for the proteins. Now, amino acids share common structural features. If we see the structure of amino acid, we can see to the carbon atom, the groups attached are COOH, carboxylic group, NH2 amino group, hydrogen, and a R group. So this is the common structural feature for all amino acids. The amino acids will differ only in this R group. Rest all the structure will be the same. So for an amino acid, there is one carboxyl end which is called C terminal and one amino end which is called N terminal. As because the carbon to which these groups are attached is the alpha carbon, hence these amino acids are known as alpha amino acids because this amino group is attached with the alpha carbon. This carbon is a chiral carbon. Why chiral carbon? Because the carbon is attached with four different atoms or group of atoms. Therefore, this carbon is a chiral carbon. So this is a common structural features shared by all the amino acids. The R group. Amino acids will differ only in the R group. This R group is responsible for the solubility of amino acids in water. Depending upon what R group is present, it will affect the solubility of amino acid in water. Whether the amino acid will be polar or non-polar, it will be soluble or non-soluble in water, is depend upon the R group present in a particular amino acid. Now, how we number or label the carbon atoms present in amino acid? This is a structure of lysine. In the structure, if you see, this is the C group, that is the alpha carbon group. This is the C atom, carbon atom, that is the alpha carbon atom, to which amino terminal, carboxyl terminal, and H is attached. This is the R group. Okay. So, carboxyl terminal is the carbon number 1, alpha carbon is the carbon number 2, and in the R group, it is carbon number 3, 4, 5, and 6. That's how it is numbered. If we want to label it as alpha, beta, gamma, then as because this carbon to which carboxyl and amino terminal is attached is the alpha carbon, so the beta, gamma, delta is in the R group. Whatever the R group is, the carbons present in the R group will take the labeling of beta, gamma, delta, and so on. So if we talk about numbering, then the carboxyl end is the carbon number 1. The alpha carbon is the carbon number 2, and from carbon number 3 it is present in the R group. Similarly, if we want to label it as alpha, beta, gamma, then the alpha carbon is the carbon to which the carboxyl group, amino group, and H is attached, and rest beta, gamma, delta is present in the R chain. So this how the carbons of an amino acid is numbered or labeled. Now come to the stereochemistry of an amino acid. If you see this structure and if you see this structure, if we want to write the mirror image of this first particular structure, what will be the mirror image? For the second structure, the image will be. So here what we can see is in the first structure, the mirror images are same. But in the second structure, the mirror images are not same. These are non-superimposable mirror images. If I place this structure over this structure, then here we'll get CH3 and H will be placed here. So these are non-imposable mirror images. But in case of first structure, it is imposable mirror images. And what we studied already in the carbohydrate videos, that is the structures which are non-superimposable are optically active molecules which are asymmetric. So if the molecule is asymmetric in nature, that means it contains chiral molecule. The images will be the non-superimposable mirror images. Hence, they can rotate the plane of polarized light, and these type of structures are known as enantiomers. So, amino acids are enantiomers. So, amino acids are optically active molecules because these are asymmetric, hence they form non-superimposable mirror images, except glycine. The first structure is the structure of glycine. In case of glycine, the R group is H. Rest, these part are same for all the amino acids. Only the amino acids differ in the R group. In case of glycine, the R group is H. So, except glycine, all other amino acids are optically active molecule because they form non-superimposable mirror images. So, these are enantiomers. These images are known as enantiomers. Now, coming to L and D, we already studied this also in the carbohydrate video. Those who have not seen the carbohydrate videos, I have provided the link at the end of this video. You can go and check them once. So coming to L and D, what we learned, the OH attached with the carbon which is farthest from the functional group, if that is on the right side, then it is called D. If that is on the left side, it is called L. So here with the simplest carbohydrate glyceraldehyde, this is on the left side. OH is on the left side. Therefore, it is an L-glyceraldehyde. For amino acids also, it is similar. Here H is, if we talk about on the basis of H, so H is in the right hand side in L form. Here also H is in the right hand side. Hence, it is also an L amino acid. For carbohydrates, we studied that D forms are mostly found in the biological system. D monosaccharides. In case of amino acids, the L amino acids are mostly found in the biological system. D forms of amino acid are present in cell wall of bacteria. Now, all amino acids derived from protein have L stereochemical configuration. Why so? It is because the active site of the enzymes, these are asymmetric, and due to which the reaction they catalyze are stereospecific. So all the amino acids derived from protein are L stereoisomers because of the enzymes. The enzymes are asymmetric, due to which the reaction they catalyze become stereospecific. Now each asymmetric center can have two possible configurations. So the molecules with n number of chiral center has 2 to the power n different possible stereoisomers. This we already studied in the carbohydrate video, how to calculate the possible stereoisomers for a given molecule. So I will provide the link at the end of this video. You can go check the video once. So now let's study about the structures of different amino acids found in a biological system. So this is the first group of amino acids which come under amino acids with non-polar side chains. So the R group present in these amino acids are of non-polar nature. Therefore, these amino acids are categorized under non-polar side chain amino acid. What are these amino acids? Glycine, alanine, proline, valine, leucine, isoleucine, methionine, phenylalanine, and tryptophan. The second group consists of polar but uncharged amino acid. These are polar but uncharged amino acids. It includes serine, threonine, cysteine, asparagine, glutamine, and tyrosine. The third group consists of polar and charged amino acid. This is the third group of polar and charged amino acid, which consists of lysine, asparagine, histidine, aspartate, and glutamate. Now, among this polar charged amino acid, this lysine, arginine, and histidine, these are positively charged amino acid, whereas aspartate and glutamate, these are negatively charged amino acid. Why so? If you can see the structures of these amino acids, we already studied that amino terminal, carboxyl terminal, and H group is common for all the amino acids. They differ only in the R group. So these all red boxes are the R groups in which the amino acids differ. Now if you see this lysine R group, it has an extra amino terminal, extra positive. Here also, it has an extra amino part. Therefore, these amino acids are positively charged because they have extra positive part. In case of aspartate and glutamate, they have an extra COO minus. So therefore, they are negatively charged. All the amino acid is having one positive end and one negative end. That is one carboxylic end and one amino terminal. Correct. So one positive and one negative, making it neutral. Now, if one extra positive is present, so the amino acid will behave as a positive. If extra negative is present, the amino acid will behave as a negative. So lysine, arginine, and histidine are positively charged amino acid, whereas aspartate and glutamate are negatively charged amino acid. Correct. Now come to the second part. If you see, these are polar, but these are uncharged because they don't have any extra positive or negative end. But these are polar. That means they can form hydrogen bond with water or they can dissolve in water. If you see these first group of amino acids which are non-polar, that means they do not dissolve in water. They cannot form hydrogen bond with water molecules. Now, what else you can see? This phenylalanine. It has a benzene ring. Tryptophan. It is also having, and from here, tyrosine is also having a benzene ring. So that means phenylalanine, tryptophan, and tyrosine. These are aromatic amino acid. Tyrosine, phenylalanine, and tryptophan are aromatic amino acids. Next, what else you can notice from these structures? The simplest is the glycine. Only H is present as a R group. So it is very flexible. Proline, it is rigid because of its structure. So in the upcoming videos, when we will study about the structure of proteins or the function of the proteins, then you have to remember the properties of these amino acids. Basically, you have to remember its structure. Not, if you don't remember the exact structure, at least you should know which amino acid is simple, which amino acid is complex, which amino acids are aromatic in nature, which amino acids are very bulky. So bulky means which have a very heavy R group: lysine, glutamine, tryptophan, tyrosine. These are very bulky amino acids. Correct. So wherever the foldings are needed, at that place the glycine should be present because it can easily fold. It is very flexible. Wherever little rigidity is needed, proline will be present. Correct. Wherever electrostatic interactions help in the stability of protein, there these positive and negatively charged amino acids will come into play. Correct. Similarly, for hydrogen bond stability, uncharged will also come into play. So, depending upon the properties of the amino acid, these amino acids are only responsible for the structure and the function of protein. So, if we want to know the structure or function of any particular protein, then we have to study its amino acid composition. If we know its amino acid composition and how these amino acids are arranged in a protein, we can easily able to detect its structure and its function. And one more point, the most abundant amino acid in a protein is leucine. This is the most abundant. And the least abundant is tryptophan. So this is in short about all the 20 amino acids found in a biological system. Next come to the amino acid derivatives. So there are many derivatives of amino acids. Derivatives means how these proteins are formed by the process of translation, right? So after translation, some amino acids are modified in a post-translation modification. These form amino acid derivatives. So let's study about few important amino acid derivatives. One derivative is 4-hydroxyproline. These are found in plant cell wall. One more is 5-hydroxylysine. 4-hydroxyproline as well as 5-hydroxylysine. These both are found in collagen, a fibrous protein. One more is 6-N-methyllysine. These are found in myosin, muscle protein. Next is 5-carboxyglutamate, found in prothrombin, blood clotting factor. One is desmosine, found in elastin. It is also a fibrous protein. One is selenocysteine. This amino acid derivative is formed during protein synthesis and not as a post-translation modification. Rest all amino acid derivatives are formed as a post-translation modification, not during the process of translation, but selenocysteine is formed during translation process only. So selenocysteine, these are also found in many proteins. Selenocysteine, that means it is cysteine only, but in cysteine, sulfur is present. Instead of sulfur, in selenocysteine, selenium is present. Next is ornithine and citrulline. These are found in the biosynthetic pathway for arginine and urea cycle. So these are the amino acid derivatives. So these are unusual amino acids which are called amino acid derivatives. These are generated during post-translation modification of amino acid residues. Now, what are the functions of amino acid? We are talking about functions of individual amino acids and not the function of proteins. So, what are the functions of amino acid? The first function is they help in the transport of nitrogen in the form of amino group. Next is they function as a metabolic fuels to provide energy. They function as a chemical messengers for communications between cells, as well as they help in cellular metabolism. Correct? So these are the functions of individual amino acids. Now, one question from my end for you all, that is, you have to tell me the non-polar amino acid can participate in which kind of non-covalent interaction for the stabilization of protein structures. I already told you that the structure or the properties of the amino acids are responsible for the structure and the function of a protein. If we know the property of an amino acid, we can easily detect what could be the structure and the function of that particular protein. Correct. So, as I told you that polar charged molecules can participate in electrostatic interaction. So, likewise, you have to tell me in what kind of interaction the non-polar amino acid can participate for the stabilization of the protein. Do let me know in the comment section. If you found the video informative, please do like, share and subscribe the channel and hit the bell icon for the next video notification. Till then, stay safe, stay happy.